this is your topic summary. So I usually don't give 40 out of 40, but this time I have given 40 out of 40. So if I see like a lot of efforts, for example, you are trying to put efforts to make really good images, making all those colors in, so that means you're putting some extra efforts there, so that means you deserve that. If you are missing few content, I should have removed the point, but this time I didn't deduct the point, but next time I'll start deducting the point because it never gave you back. So make sure you don't miss any point. And so I think you all have worked really hard on this. It's not like no one has worked more or less, but only a little more effort would have make it from 38 to 40. So again, those we can go up from here, and we need some subheadings also. This is actually this. You got all the things, right?
Those are all my rough drafts, I think.
This is your rough draft? I don't know.
I have it, it was the test design.
I think there was something which was kind of confusing to me.
She put that, she edited it, that's why her name is also on my paper.
So everyone got all the three?
Yes.
Okay. I was kind of confused, like what I'm getting based on that. Okay, so everyone can improve if you're already there, but you can also go down, so make sure you're putting the efforts there, right? So you can go down and improve, both things can happen. Okay, now, so we will be meeting tomorrow at 1, not 12:30. You have a meeting which will end at around 12:50 something. So we'll try to make it at 1 o'clock. Now, how much time it took us yesterday? Do you have any idea?
I think like 2 hours in the lab.
2 hours in the lab. Yeah,
If we don't, like, if we're doing some of the same stuff, it should go a little faster, so I think about 2 hours for some.
So we will be doing almost the same stuff. What we'll be doing, we will have those cells in the flask. Now you know how to do it, right? I didn't have to show you. Remove media, do drips in, start new flask, count cell, plate cells. So I think that should be fast. So now we can make 2 groups. We have 2 flasks going on, so you guys can make 2 groups. So total we'll make 6 of the petri dishes, right? And yeah, that should be good. So only time it will take us maybe 5-10 extra minutes. No, we have drips in thought already there. Yeah, we should be okay. Yeah, should be okay. Okay, so we will finish this topic, and this will be it will not take more than 30-40 minutes to finish it. And then you guys can tell me whether you want to start on Friday. It's up to you. So you guys need to tell me you want to start on Friday or you want to start the new topic on Monday. You guys can take time and decide and let me know. Now we are on this thing. What we are saying is the fusion of genetic material. And again, again, we are talking about mammals, so it will be slightly different than it was in the CLG. So, the male sperm, oh, I need to ask you, what were you doing last time? Before my first, okay.
So we started with mechanism of capacitation at the tail,
Uh-huh.
and we had first that the removal of cholesterol activates the bicarbonate channels on the tail, and then we have, once this happens, the bicarbonate ion gets into the cell, increases the alkalinity, and then we have, album, cholesterol, right?
Mhm.
And then potassium ion, potassium channel opens and potassium goes out of the sperm. And then these two, and then we have the voltage gated calcium channel, and this opens to bring in more calcium.
Why do you call it voltage gated?
Because it's opens due to the alkalinity. So the
Well, couldn't be as much of a voltage. Then should be,
potential is.
then should be acid-base gated. Why we say voltage gated? So if potassium ion are going out, voltage of the membrane is going to change, so they can sense that voltage change. They
Right,
can-
the potential. of-
Yeah.
Okay.
Yeah.
Right. And so once that potential changes, then voltage gated calcium channel opens, increases calcium, and then that activates diamine ATPase and the mitochondrial ATP production, makes the sperm more hyperactive, and then this calcium along with the bicarbonate will activate sperm individual cyclase, SASE, and then that creates ATP, or, it uses ATP to create cyclic AMP.
Okay, then what happens?
So then we talked about the capacitation of the head, so the same things that happen.
No, we are still in the tail. Just talking about activation of SASE.
Oh, okay. Activation of SASE, that converts ATP to CAMP. CAMP activates protein kinase. The protein kinase phosphorylates the tyrosine kinase, and that phosphorylates other sperm proteins. The calcium activates the diamine, which is ATPase, and it activates mitochondrial ATP production. And together, the tyrosine kinase and the ATP production activates the sperm, and that makes the tail hyperactive and then release from the wall.
Release from?
The
Isthmus, okay. Then what happens?
And then we went to the head, so then, it goes through the same process of opening the bicarbonate channels and the potassium channels, and then the voltage gate, or increases the alkalinity, and then the voltage gated calcium channels open, and then, the bicarbonate and, calcium activate, SASE, is that how you said it?
Yeah. Sperm adenylyl cyclase.
Yeah. And then, so that converts ATP to CAMP, and CAMP activates protein kinase A, which phosphorylates the protein izumo in this case. And then when izumo is phosphorylated, it moves to the acrosomal vesicle membrane. And this is critical for the sperm membrane to bind to the egg membrane. And then we also talked about how having high calcium levels, high CAMP levels, and high bicarbonate levels are also important because they also help in the facilitation of membrane fusion.
Okay. Then?
So once the sperm is capacitated and it leaves the isthmus where it goes, it uses heat as a cue because it can sense temperature differences of up to 0.014 degrees Celsius, and the isthmus is 2 degrees colder than the ampulla, so it follows that temperature change. And then once it gets to the ampulla, it senses the chemotaxis released from the egg, and the cumulus cells around the egg secrete progesterone, which can attract the capacitated cell. or
Secrete
sperm.
what?
What?
What the secrete?
The cumulus cells secrete progesterone.
Progesterone, okay.
And then those can attract capacitated sperms, and when they bind To the sperm's calcium channels, they open it only if there's bicarbonate present as well, which is when it's capacitated. So it doesn't do it to uncapacitated sperm. And then that causes entry of more calcium into the cell, and then more ATP and dynein activation, which leads to hyperactive sperm in the oocyte's vicinity. The cumulus cells then secrete CRISP-1 in the inner layer, and this acts as like a chemottraction as well, and it causes the sperm to move into the inner layers and then the zone of use.
Okay, then what happens?
Then we talk about the acrosomal reaction and recognition of the zona follicula. So the acrosome reaction occurs in the cumulus cells, and then on the zona follicula there are 4 different types of zona proteins, ZP1 through 4, and the acrosomal process binds to ZP2, and acrosomal fusion releases contents which help clear the cumulus cells, and late arriving sperm bind to ZP3. And then there's no fast block to polyspermy, but the cortical granule reaction happens, and ovastatin clips off ZP2 for no new sperm can bind. And then the Zuno, so the Zumo on the sperm bind to the Zuno protein on the egg membrane, and they recruit CD9, which aids in membrane fusion. And once first Zuno binding, all other Zuno proteins move to the cytosol.
So that prevent, again, polyspermy, right? There was one question, I think Rachel, that was your question. The question was, as I remember, was you said when the mature follicle ruptures, then we said that corpus luteum produces progesterone, right? That was your question?
Mhm.
Can you repeat your question again so that everyone can?
So, whenever the follicle ruptures, it produces a lot of progesterone, so is the sperm ever attracted to that and not the egg?
Okay,
And then
that's
what
it.
mechanisms-
You're understanding the question.
Yeah.
What you think, why that doesn't happen?
Isn't it left in the ovary?
Hmm?
Isn't it
Because
left-
that happens in ovary.
Oh, okay.
So ovary and fallopian tube are separate organs, right? So that doesn't happen. So, but remember we said the progesterone release prepare uterus for implantation or pregnancy. But how that happens? Because that is released in the blood. That's not direct release. So here, it's direct release in that tube, and then it basically attracts there. That is making sense? So otherwise, what is the purpose of rheotaxis? That's what you said? That was your question, right?
That was my question.
That was your question, okay. So rheotaxis means that's kind of a direction cue where sperm need to go. So sperm need to move in the opposite direction of the flow.
Okay.
So that's another direction cue. We said many direction cues are there, so that is one of the direction cue. The sperm is going to move opposite to the direction of the flow. Was there any other question which I missed? Okay. So then we are saying that now the fertilization has taken place. Now we are saying that fusion of genetic material should take place. Okay? And the male sperm, it carries a pronucleus, and we know that pronucleus is haploid. Right? In the oocyte, the pronucleus is still in second meiotic metaphase. Right? So then how, so we said this is arrested there, right? So that arrest should go away. And also, this pronucleus, which is a haploid, but we know that it has very compact chromatin, should also get ready for the fusion, right? So what happens that we know that in the haploid male pronuclei, the chromatin is tightly bound, and it is bound with the protamides. Protamides, remember they replace histones, and it's tightly bound. Now these proteins are actually, they are tightly compacted by disulfide bonds. And that is the bond, right? SS bond is disulfide bond. So this bond need to be reduced, okay? And you remember in the egg, we have glutathione. So that glutathione which is present in the egg, what that can do, that reduces disulfide bonds and it changes to SH, and this is SH. So this bond is broken. Okay, so that bond is now broken. So that means now the proteins can be released out. So proteins are released out. So that leads to release of proteins. Therefore, chromatin loosens up. So chromatin is now free. Now, the important thing is, this is still happening in the nucleus. So still there is intact nuclear envelope. Because this thing is happening right here. So male sperm, male nuclei, male pronuclei enter in the egg, and because those are nuclear pore, they can help the GSA, the glutathione to enter, and glutathione reduce disulfide bond, and that loosen up the chromatin. That's the first step that is going to happen there. Now, the other thing is going to happen is, remember, whenever we have the egg fusion with the egg membrane fuses with the sperm membrane, we know that calcium influx takes place, right? So under the influx of calcium, so we'll have high calcium. High calcium is there always. And when that high calcium is there, that is going to do multiple things there. Number one, is going to inactivate MAP kinase, right? So if MAP kinase is inactivated, what is going to happen? That is going to start DNA synthesis. Now this DNA synthesis occurs separately in male and female nucleus. So separate, because they still have their membrane attached to them. Okay. Now, what happened that, remember that female nuclei is still in second meiotic metaphase, right? So now DNA synthesis has occurred. Then what happened that there is a factor, we said the, remember, if you remember, we said cytostatic factor are there. Those cytostatic factor, they were the inhibitory factor. If you remember from previous notes, they were the inhibitory factors for meiotic promotion. They were inhibiting that. So it means we need to degrade this cytostatic factor, right? And that happened because these calcium activity, calcium, high calcium, what it does, it binds to a protein And that protein name is called calmodulin. Calmodulin, okay? And that calmodulin, once calcium is bound to this calmodulin, this calmodulin activate calmodulin dependent kinase. Calmodulin dependent kinase is activated. And when that happens, what that does, that phosphorylate cytostatic factor. Okay. So once that cytostatic factor is phosphorylated, this phosphorylated cytostatic factor degrades. So that's the first thing happening. Okay, so cytostatic factor is now degraded. Now the second thing is going to happen there is, so we have talked about two things. Two things have happened. Inhibition of MAP kinase have taken place, that have started the DNA synthesis. Second thing, we said that cytostatic factor, which was inhibitor of the mitosis, that has been degraded, so that barrier is also gone. Now the third thing that happened is, so this is the first thing, start of DNA synthesis. Second is this. And the third thing, what is happening there? Third thing is happening, if you remember from your previous classes, so we said so this is the chromatin, right? And this is the sister chromatin. Sister chromatin, right? This happened in the metaphase, we have like this. Now they're going to separate in the anaphase, right? Remember that these two are actually bound together like this. These are stayed together. They are secured together by a protein called securin. Securin protein is there. So securin keep those sister chromatids together. Okay. So what happened? That all these activities. So the third step happened that this securin is degraded. So securin is degraded. That's the third step. Now securin is also gone. So that means this is going to lead to, so that is going to lead to now these chromatids are now free to separate out. Okay? Then what happens? So all these things are happening because of that calmodulin dependent kinase activation. That is triggering all those things. Okay? Now the fourth thing happened there. Cyclin D is degraded. So cyclin D was kind of keeping it on that place, because remember the cyclin dependent kinases are important. When this cyclin is going, so that cyclin which was keeping it in that phase is gone, so that next cyclin dependent kinase can activate and go to the next step. Remember that second meiotic division is essentially a mitosis division. In the first is a reductional, you remember that, right? And the second is kind of mitosis. So cyclin D degraded. So four things are happening there. More DNA synthesis, degradation of cytostatic factor, degradation of securin, and degradation of cyclin D. And when that happens, all these four things lead to resumption of second meiotic metaphase. So if I have to draw it like this is suppose your egg is there. So here is the male pronuclei. So this is the new pronucleus from sperm. Right here, somewhere the second division start like this, and that lead to formation of second polar body, and this is the female pronuclei. Making sense? Or no?
Can you just say the parts of it again, of the
Okay,
drawing
let
again?
me, this is clear, right? So I can erase it, right? So I'll draw it a little bit. So if I have to draw this, a bigger. So now the fusion has taken place. So right here is this is your centriole or centrosome or centriole from sperm. Right? That should come from sperm. Then this is the cell membrane, like this. And this is the male or sperm pronuclear. Right? Now, when this has entered, the disulfide has been reduced and more DNA synthesis has started here. Same way, the same thing happened right here in the female nucleus also, right? But that female nucleus was stuck here in so this was the second meiotic metaphase nuclei, right? And that was still 2N. Remember, that was still 2N. Now what happened here? When this thing starts, we said all these barriers are removed. The cytostatic factor has been degraded, right? We said that securin has been degraded, and we said cyclin D is degraded. So that means it can complete its second meiotic metaphase. So what is going to happen? This is going to divide in a way that this nucleus is going to divide in two, and then you will have a small cell is going to be produced there. All the cytoplasm will be retained by this nucleus, and this will be our second polar body formation. So now after this division, this is the haploid female or egg pronuclei. That is making sense? Now what is happening there? And we know this will finally degenerate, right? This will not separate, this will degenerate. Now what is going to happen? So this is going to degenerate. And then there is nuclear membrane here surrounding this nuclei. Surrounding this nuclei also there is nuclear membrane. Surrounding this nuclei also there is nuclear membrane. And there is genetic material inside. So what happens? This centriole, which was right here, so remember this centriole is going to divide into two. So you will have two centriole. The sperm centriole divide, give the female, because there is no centriole in the egg, right? So this divide, when this divide, this will start producing estrus and start moving in the other direction. Okay? And when they will move in the opposite direction, what they are going to do, they are going to make more and more these microtubule. right? We know that thing, that phenomenon, how they make microtubule and all those things. Yes or no? So what happened? These microtubules, which are there, they will help these two nuclei to move toward each other. So this nuclei will move toward this is going to move toward this. So they will move toward each other, okay? And what is going to happen when they're going to move toward each other? So ultimately what is going to happen, this is one, this is other, one nuclei, this is the other nuclei. So they come close to each other. This is going to happen, they're going to come close to each other. When they come close to each other, so these nuclei, the pronuclei, they migrate toward each other. When they reach close to each other, at this point, the nuclear membrane disintegrate. So when they come close to each other, this nuclear membrane is going to go away. So in a way, if I'm drawing here, so you will have all the chromosomes from male and female together, and there is no nuclear membrane right there. This is a diploid cell being formed, right? Now, it never stays in that position. Immediately, when this thing happened, immediately when this nuclear membrane goes away, the first cleavage starts. The first cleavage starts. That means the development starts. Making sense? So what we said? We are saying that when the female and male, when the fertilization takes place, when the sperm and egg fusion takes place, that leads to entry of male pronuclei in the egg, or oocyte. And we are saying that the first thing that is going to happen is you need to loosen the chromatin, the tightly compact chromatin of the male pronuclei. And we said that is tightly compact because we have the protoamines, and those protoamines make it more tightly compact because those protoamines are bound together by the disulfide bond. And then there is glutathione in the egg cytoplasm that reduce those disulfide bonds, and basically the protoamines can loosen up and they can go away. And then the chromatin will loosen up, and we know that protoamine will be exchanged by the histone, and histones will also be made in the egg, right? So now that is loosened up. Then we have high calcium. Calcium inhibits MAP kinase, and when calcium inhibits MAP kinase, that take away the inhibition of the DNA synthesis. So DNA synthesis start occurring in both male and female pronuclei. Then we are saying more calcium is going to activate the protein called calmodulin, that is a calcium binding protein. When that protein is there, then calmodulin kinase gets activated. When calmodulin kinase gets activated, that's going to phosphorylate cytostatic factor, and once cytostatic factor is phosphorylated, that is degraded. So that means inhibition of meiosis promotion is gone. And then we are saying the second thing that is going to happen is you are going to take care of this cyclin D, and that cyclin D is also degraded. That's the second inhibition that is also gone. Third is you want to make sure that these chromatin can separate from each other. So the securin is also degraded. So that means once that is degraded, so female nuclei is now ready for the second meiotic metaphase progression, and that divide, and you get a polar body, and you get a small pronucleus there, right? So now you have the male pronucleus and the female pronucleus both ready to combine each other. Then, in the meanwhile, these centriole divide into and start making the microtubules, the spindle fiber. They help these pronuclei, male and female pronuclei, to move toward each other. When they still have the nuclear membrane, but when they are in close contact with each other, nuclear membrane disintegrate, and now they are free chromatin. Now the cell has become actually diploid cell. Because two nuclei, they have separate, so now the chromatin is right there. And then immediately the first division start, actually the cell division starts, so the new organ will start forming. Making sense? So talk to each other for a second, then we'll go.
The proteomides are tightly compact by disulfide. And the eggs have glutathione, which reduces disulfide bonds, which is how the bonds are broken for proteomides to become released and release and spread from food. High glycine levels inactivates MAP kinase, which starts DNA synthesis and occurs separately in male and female babies. High calcium levels also bind to a protein called calcium, whatever, and it activates it. And then that phosphorylates the cytostatic factors, and the phosphorylates the cytostatic factors degrade. And we said that citron also degrades. Then once those four things are started or done or whatever, it leads to the second meiotic benefit. So we did some drawings. So it makes the second polar body, and then the centriole splits, and it starts going to the opposite poles, and then pushing the sperm and egg together, and then once they push together, their membranes degrade, and then the first cleavage starts.
Any doubt here? Any doubt? Okay, now. So we are talking about activation of mammalian egg. So essential mechanism is same as it was in sea urchin, right? How that happened, we said we have calcium, and that calcium has some early response, some late response. The same way here the calcium plays a critical role. So rising calcium activates egg metabolism. But here is one important difference. In the sea urchin, what we saw, for example, calcium level was initially this, and then we said it goes like this, and then it comes back later, right? It was like that. A single peak in calcium goes down. In mammals, you get waves of calcium spikes. So calcium will be like this, go up, go down. It will go down, go up, go down, go up. And you see here that there are waves of calcium spikes, but each spike is of different amplitude and different duration. So spikes of calcium take place, waves of calcium take place. Calcium goes up, goes down, goes up, goes down. So there are series of calcium spikes or calcium waves. But each wave is different in amplitude, different in duration, and of course they are occurring at different times. Okay? So now, what caused the calcium release? That is the major difference. Okay? And the other major difference is what caused calcium release. Of course, throughout the animal kingdom, IP3 is the main releasing factor for the calcium. And we know that IP3 is produced from PIP2, and we know that we need the enzyme PLC, phospholipase C, right? Now, when we're talking about that thing, we said, hey, there is kind of a debate whether the calcium releasing factor is coming from the sperm itself, or it is only the binding of the sperm to the egg membrane induces the PLC activation in the sperm membrane, right? And we also said that this NAAD is also present in sperm, and we said that also can release calcium, right? But what they have found in the mammalian sperm, so mammalian sperm contain itself a PLC, phospholipase C, that is present in the mammalian sperm. And that particular form of PLC is called PLC zeta. So mammalian sperm contain PLC zeta. And that PLC zeta can act on PIP2 and produce IP3. There it was PLC gamma, right? So you have a PLC form, phospholipase C, a zeta form is present in the sperm. So sperm carries it with itself. And that people know when they do the intracytoplasmic injection during in vitro fertilization. So some sperms, they fail to initiate the egg activation. And when they look at those sperm which fail to initiate egg activation, they said they were devoid of PLC zeta. They didn't have PLC zeta. So it is now clear that PLC zeta comes from sperm in mammals, and that utilizes PIP2 to make IP3. And that IP3 is responsible for calcium release from endoplasmic reticulum. Now here is other thing. So if we are saying that there is a wave of calcium, so calcium goes up, calcium goes down, okay? So where that calcium goes when it goes down? So that calcium coming from ER, released from ER, and then goes back to ER. Now there's another thing. The calcium which is produced from the ER is not sufficient enough for the egg activation in mammals. So in sea urchin we said all the calcium is coming from the ER. But here the demand of calcium is too high. So that means that extracellular calcium also required. It means the calcium channel which are on the cell membrane, they are also playing an important role. So they also open up and calcium gets in. Now calcium is critical if you inhibit extracellular calcium. So the female pronuclei fails to undergo meiosis. So then the resulting embryo will be haploid, diploid, or triploid? That will be triploid, because you will have diploid female nuclei, and then you will have haploid male nuclei coming there, making it triploid. Okay? So the same thing happens here. What is happening there? What caused the, you have high calcium activation, waves of calcium activation, but ultimate result is same. Number one, you will have cortical granule reaction. Number two, you will have synthesis of DNA, and that will lead to resumption of cell division. And then you will have translation of maternally stored mRNA. Now, all these events require different level, different timing of the calcium. That's why in mammals, that's why you have the oscillations there. So making sense? So we are saying that in mammal, the activation of the egg is through calcium, like in all the other organisms. But here, the calcium does not enter and just goes out. Here, calcium elevation takes place in the waves. And each of the waves differs in amplitude, differs in timing, and differs in duration, right? And then we are saying the calcium which is coming here is released by the activation of, by the production of IP3. That IP3 we know is produced by PIP2, but in this case, the enzyme which is missing is coming from the sperm itself. So sperm carry that enzyme with it that is going to act on PIP2 to make IP3. And that enzyme which is present in the sperm is called PLC-zeta. PLC-zeta enters the egg. And then we give the example that in vitro fertilization, when you have direct transfer of sperm in the egg, that induces the egg activation because of the PLC-zeta. And the sperm which are devoid of PLC-zeta cannot activate the egg. Then we are saying that all of these events, calcium release, then calcium goes back, that get back to the ER. But we are saying that ER calcium is not sufficient for the mammalian egg activation. You need calcium from outside as well. If you inhibit the calcium coming from outside, it cannot proceed to the meiosis, and you will get a diploid egg, and the resulting embryo will be a triploid. That's going to die, of course. And then we are saying the same thing as we said in the sea urchin. Three main events are going to happen. Cortical granule reaction happen, and then you have synthesis of DNA and resumption of cell division, and finally translation of maternally stored mRNA. That is making sense. So guys can go over this, then we'll go to our next topic.
Raising calcium levels at and that goes in a series of ways. Calcium spikes, all of different amplitude and duration. Causes this release is IP3 because the mammalian sperm contains PLC-zeta, which is needed for sperm active or fertilization activation. And PLC-zeta converts PIP2 to IP3, which then causes the release of the ER of calcium. The oscillations, when it spikes, is the release of calcium from ER, and when it goes back down, it goes back to ER. And then because this is a high enough level of calcium, extracellular calcium is also needed. And if inhibited, like female programs, like go to endomeiosis, which cause like triploid, and two end up in sperm. The lack of calcium causes a cortical granule reaction, DNA synthesis, and modulation of maternally stored mRNA. And all these processes require different levels of calcium. That's why osmosis.
Confusing? No, you guys are good? You guys are fine? Okay, now. So now we are on the next subtopic, and that is non-equilibrium valence of mammalian pronuclei. So, what does it mean? It means that pronuclei are not similar. Why they are not similar? They have the half of the set of the chromosome. That means they have half of the genes which are required. But then you will say, okay, if that is true, then either only having the male pronuclei, instead of one pronuclei, you get two pronuclei, that will give you a perfect human. Because same number of chromosomes, right? Same genes. Male and female have same genes, right? Except the sex chromosomes. Then you will say, okay, if we have the same female there, if you have diploid female nuclei, that should give you a new individual. Like we did give the example of silver salamander, right? We said the silver salamander has diploid nuclei. They just need Jefferson for stimulation, and then they give the whole all females. That should happen here. You should have all male or all female production, right? But what happens? For example, if you take a female, not a female, an egg, okay, and egg should have a pronucleus right here, and you remove this pronucleus. So now you have removed the pronucleus, so that means you have an egg. That egg is now completely devoid of a pronucleus. Okay? Now what you do? You fertilize it with a sperm. So you do the experiment, you fertilize it with a sperm. What is going to happen is that sperm pronuclei is going to go there, right? This sperm pronuclei is haploid, right? We know that sperm pronuclei is haploid. So sperm pronuclei, what is going to happen when this is going to happen? That sperm pronuclei will divide, and because everything is activated in the egg, metabolism is activated, right? Metabolism is activated, so that means the cell division should take place, everything should take place. So what is going to happen, this sperm pronuclei will divide and give a diploid number of chromosomes. You will say now you have a diploid number of chromosomes, so we are good. Okay? So now it becomes, changes to, so this is N, now N changes to 2N. Okay? Now these cells, they survive. They survive, they divide actually. Multiple cells are formed. But they just stop there. They form a tumor. They form a tumor, and that tumor actually resembles placenta. And that tumor is called hydatidiform mole. That causes hydatidiform mole. So the cells survive, cell divide, but they don't form any organ system. They just form a tumor. That tumor looks more like a placenta than anything else. So that means male only cannot do this. Okay? Then they say, okay, let's do the experiment with the female one. And you have this egg, And you know that if we know that this division, the pronuclear division depend upon the calcium, right? So if you remove the calcium, it's not going to divide, it's going to stay diploid. Making sense? So take out the calcium, keep it diploid. Now you have a 2N female. This is oocyte, right? And this we did not let it divide by removing the calcium. Now what happens? Then again, you know that if you add PLC zeta, go more calcium, everything will just activate, right? You take a PLC zeta, inject the pill, everything is IP3, right? You put that, it will start dividing. So say you put an injection of IP3. That's a lot of IP3, put in the area where the calcium is. Now rest of the thing, egg activation starts. So egg is activated. This was done in the mouse, okay? In the mouse, the gestation period of mice is 18 days. Gestation period. Mice, okay? What they found, that this actually divide. This not only divide, what it does, it forms organs, including spinal cord, heart, and that heart is actually beating. A beating heart. Now, all those things happen, but they survive, they go up to 10 days. So it starts developing, goes up to 10 days. And after 10 days, what happened? This actually died. After 10 days, embryo died. So think about in terms of human, if a 9-month gestation period, so almost 5 months, right? So it develop, every organ system is formed. So then it dies. So what we understand from here, we are saying that, okay, if you have only male nuclei, it doesn't form anything. If we have female nuclei, it goes up to here. Same gene, everything is same, right? It means that male and female, the genome should be different in epigenetic patterns. And what they have found, almost 100 genes are methylated in male and female specific patterns. So methylation patterns are critical. So what if you change the methylation pattern? What is going to happen? So they did the experiments. They changed the methylation pattern. They said, okay, once this nuclei divide, take one nuclei, change the methylation pattern. When they did that, they were successfully able to produce the new embryo. So, remember, in the beginning we said, when they come from primordial germ cell, we said when they're moving in, their methylation pattern are completely removed. Then we said when they move from there to the germline cell, we said male and female specific methylation patterns are formed, right? So now when they're meeting together here, so you need both type of pattern to start a new development in the beginning. But later we'll talk more how that changes further. So if that methylation pattern is not there, you cannot have a complete, fully formed individual. That's why we said that male and female plurinuclei, they are actually non-equivalent. they are not equal in terms of their genetic gene expressivity. Making sense? So talk to each other and then.
So this is all like experimental, like it's not something that happens.
Well, if you don't have, if it doesn't happen, it doesn't matter.
I don't know.
So you remove the female pronucleus, but then let the sperm fertilize the egg, the sperm pronucleus will divide. Give a diploid number of enough, or has enough number of diploid. Then it splits the cell, then it forms a tumor, or placenta, which is called cytoplasm. Or more, something. Developed for 10 days and then it died. And then there are 100 genes. 100 genes are have methylation patterns, specific patterns. They're different between male and female. That's what I got.
Yeah, but it is up to 100 genes, or about 100 genes methylation male and female specific factors.
Okay, everything is clear here? Any question? Okay, so we are done with this. Now, tell me what you want. You want to start meeting on the Friday or you want to wait till Monday? Talk to each other for a minute and let me know.
Well, we won't be here on Friday, so.
So definitely